Assalamualaikum warahmatullahi wabarakatuh. Good morning, class. How are you today? I hope you all are doing fine today. Nice to meet you again in this biology class. But before we start our lesson today, let's pray first. Pray start. Finish. Now I will check your attendance. Please raise your hand after I call your name. Okay, class. In our last meeting, we have already talked a little about enzymes. And today I will explain how enzymes work and factors that affect enzymes. Okay, now we are going to take a look what enzymes do and the differences between log and key model and induced feed model of enzyme action. Firstly, we need to know that a living cell, to function properly, it has to carry out a lot of chemical reactions every day. But the problem with this is most chemical reactions naturally quite slow. To make speed up reactions is by increasing temperature, but this way can cause a few problems. One, it can require a lot of energy to keep an organism warm. Two, high temperatures can damage our cells. And three, higher temperatures also can speed up all of non-useful reactions that we don't actually want. A better way to increase the speed of chemical reaction is with catalyst. We can define a catalyst as a substance which increases the speed of reactions without being changed or used up in the process. It means the catalyst can be used over and over again without being used up. Enzyme is a particular type of catalyst that is made by living organisms. So we can call enzyme biological catalyst. Enzymes basically large proteins. So like all proteins, enzyme made up of long chains of amino acids. There are two models of enzyme action that you need to know about: original log and key model and induced feed model. First, in log and key model, scientists thought those substrates had to fit perfectly into the active site just like a key fits perfectly into a log. And then we need to know that the enzyme actually changed shape slightly so that it fits more neatly around the substrate. This idea that the active site changed slightly to perfectly fit the substrate is known as the induced feed model. Enzyme can speed up reaction because they have an active site, which is a region on enzyme that have unique shape and specific to the substrate of the reaction. And the unique shape is really important because if the substrate doesn't fit the active site, then the reaction won't be catalyzed. Okay, now we need to look at how enzyme works. If you think about chemical reactions, they often involve taking one thing, which we call a substrate, and breaking it into multiple parts, which we can call products. Or it could be the reverse, where multiple substrates are combined to make a single product. Now we will consider how temperature and pH affect the enzyme. Let's start with temperature. So this is a graph that shows how the rate of an enzyme control reaction changes with temperature. We can see that a temperature increases, so the rate increases too. But after 37 degrees, the rate starts to drop rapidly. This is because the high temperatures start to break some of the bond holding the enzymes together. So the active site starts to change the shape. If the active site changes the shape too much, the enzyme won't be able to bind the substrate and catalyze the reactions anymore. At this point, the enzyme has been denatured. The temperature which in enzyme function best is known as optimum temperature. So the optimum temperature of this enzyme will be 37 degrees because that is the temperature made the rate peaks. In human body, it's generally kept around 37 degrees. It's a pretty common optimum temperatures for human enzymes. But for different enzymes can have different optimum temperatures too. Now, if you remember from chemistry, pH is a measure of acidity. And now we can see at this graph. From this graph, if the pH is too high or too low, it can lower the rate for reaction. Just like in temperatures, this happens because the enzymes start to break, so the active site changes the shape. As you know before, if the active site changes the shape too much, the substrate cannot fit at all, and the enzyme can be denatured. The optimal pH will depend on where the enzyme normally works. For example, most of enzymes in our body work at neutral pH around 7, but the enzymes that work in stomach have an optimal pH around 2. It goes pH of the stomach. Okay, now I will give you an example how enzyme works. You have to try this at home later. First, take a piece of bread and chew it for a while. Pay attention how the flavor of the bread changes. After a while, it should start tasting sweet. But why does this happen? Enzyme is the answer. The main component of bread is starch, which is large molecule that is tasteless, and our saliva contains of amylase, an enzyme that can break starch into sugar. So that's why after a while the bread starts tasting sweet. Okay, I think it's enough for today, but before we end this meeting, I wanna ask some questions. First, from this picture, can you determine if this clock and key model or induced fit model? Yep, that was right. This is induced fit model. This model suggests that an enzyme, when binding with its substrate, optimizes the interface through physical interactions to form the final complex structure. And the last question is, from this graph, can you guess the optimum temperature for this enzyme? Yes, 40 degrees Celsius is the optimum temperature for this enzyme. Okay, class, that's all for today. Please read again your textbook and don't forget to try at home how enzymes work by chew the bread. Thank you for your participation and have a nice day. Wassalamualaikum warahmatullahi wabarakatuh.